Welcome guys to RSE level. Today we'll be looking at stem cells. So what is a stem cell? A stem cell is an undifferentiated cell which divides to give rise to other types. So for example, stem cells, which can be drawn which is just a cell, can be differentiated to form skin cells. It can be differentiated to form liver cells. It can be differentiated to form uh, cells that make up the lining of your intestine or goblet cells. Basically, it can divide to give rise to all to any of the 216 human cell types. Now, there are three main types of stem cells. There is the totipotent cells, there is the pluripotent cells, and there is the multipotent cells. Now, we will define each of them. So, totipotent cells, stem cells. These are stem cells that give rise to all 216 cell types. The exact definition of a totipotent stem cell is a stem cell that gives rise to all cell types, including extra embryonic cells. So it includes extra embryonic cells and gives rise to all cell types. All 216 human cell types. Now, what are extra embryonic cells? Extra embryonic structures are basically the yolk sac, the yolk, placenta, umbilical cord. Those are a few of them. So these are extra embryonic cells. Embryonic structures, sorry. Totipotent stem cells will give rise to form all 216 cell types, including the extra embryonic cells. Now, it is worth noting that when fertilization occurs, as shown here, when the sperm and the egg fuse, a zygote will be formed, and that zygote is let to grow for 4 days or 4 cell divisions. Now, usually a zygote will go through a mitotic division to form 2 identical cells. This is the first mitotic division. Then it will divide again, second mitotic division to form 4 identical cells, and the third to give 8 identical cells, and on the fourth mitotic division it will give rise to 16 cells. How many are these? 14, 15, 16. So it will give rise to 16 genetically identical cells. What is this structure called now? This structure is known as the morula. A morula is a solid ball of genetically identical totipotent stem cells. It's usually made up of 16, or if one more mitotic division is allowed, it will give 32 cells. So it's 16 to 32 cells, all of which are totipotent stem cells. Now, usually when doctors or researchers want to obtain totipotent stem cells, they will harvest it from IVF and spare embryos after they let it grow for a couple of days, usually for 3 to 4 days after fertilization. The second stem cell we'll be looking at is the second type is a pluripotent stem cell. Stem cell. A pluripotent stem cell is basically it's a stem cell that gives rise to most cell types. So it gives rise to most cell types, but it doesn't give rise to extra embryonic extra embryonic structures or cells. And we looked at this. This is the yolk sac, the yolk sac, the placenta, the umbilical cord. Now, it is worth note. Let's go back to the original diagram. So, when a zygote will continuously divide, it will form a morula, as shown here. That morula will divide further to form a blastocyst. We'll also be looking at this structure, which is known as the blastocyst. Now, the blastocyst is made up of the inner cell mass, which is this. It is also made up of this part, which is known as the trophoblast cells. This is too big. So this is known as the trophoblast cells. What are the trophoblast cells? We will be looking at that actually, but let's complete the labeling process. Now, this region here that I've shaded is known as the blastocele. The blastocele. It's also known as the fluid cavity, and the inner cell mass, which we have labeled and it's in blue color, is it's basically what forms the embryo. So this will become the embryo, and the outer cell, which is this region, which is known as the trophoblast cells, will form the placenta. And there is also a blastocyst we'll define it here. Let's just raise the font size. Okay, so a blastocyst. Oh yeah, it is also worth noting that this center region, the inner cell mass, is the pluripotent stem cells. It's what makes up the pluripotent stem cells. And we will now define a blastocyst. A blastocyst is a hollow ball. It's like, instead of the morula, which is a solid ball, the blastocyst is a hollow ball that contains 70 to 300 cells. So we can just write that, hollow ball, 70 to 300 cells. And the inner cell mass is this inner cell mass cells are pluripotent. So it's the same process when scientists or doctors or researchers want to extract these pluripotent stem cells. They will let IVF occur in the laboratory. Then, using the spare embryos, they will let the zygote grow for a couple of days, and this will take actually longer. So 5 days instead of the 3 to 4 days for totipotent stem cells. Instead, it will take 5 days by mitosis, and after 5 days, a blastocyst is formed. So from a morula, which is 3 to 4 days, on the 5th day, a blastocyst will form, which contains pluripotent stem cells inside, which is the inner cell mass cells. It is also drawn over here, embryonic stem cells. And the last cell type that we will look at is multipotent stem cells. Multipotent stem cells, unlike pluripotent stem cells and totipotent stem cells, they actually don't give rise to that many cell types. So they give rise to few cell types. And it's also worth noting that the source of these stem cells, unlike the previous two sources, which were for totipotent stem cells, were the morula, and for pluripotent stem cells was the blastocyst. For multipotent stem cells, the source is actually adult stem cells or somatic stem cells. So source is adult or somatic stem cells. We also have to, and the types of these are, the types of adult or somatic stem cells are bone marrow. This gives rise to white blood cells. There is also skin stem cells. There is brain stem cells, and there is liver stem cells, to name a few. Now, what are adult stem cells? Adult stem cells are basically undifferentiated cells that are found amongst differentiated cells in a tissue or an organ. So there is a big difference between normal stem cells and adult stem cells. It is worth noting, and it could be asked, adult stem cells are undifferentiated cells like stem cells. However, they are found amongst differentiated cells, and they're usually found in organs or tissues of the human body or other animals. And the purpose of an adult stem cell is for repairing of tissues, like we can see here. So these are some of the somatic stem cells that we can see: cardiac stem cells, neural stem cells, and there are more. But basically, the use of these stem cells are for repair and growth of the tissues in which they are found in. Now, there are two definitions that are worth noting. There is cell determination and cell differentiation. Determination is, let's just write that down. So, cell determination. Cell determination is the process by which an undifferentiated cell, an undifferentiated stem cell, will actually begin the process of differentiation to form a particular or a specific cell with a specific function. Now, it is worth noting, once a cell decides that it will become, for example, a liver cell, it will end up becoming a liver cell. That is known as cell determination. Once the stem cell decides what it wants to be, it will become that cell type. There's also cell differentiation, which is quite different from cell determination, although they follow the same parts. So cell differentiation is the process by which animal cells become specialized for a particular function and lose the ability to form other different types. So it's actually taken a part of cell determination, but it's taken that process. Now, let's look at that same example we've drawn above to become a liver cell. Now, in that process, which is the center region, what occurs here between the transition from a stem cell to a liver cell, a specific liver cell, the undifferentiated cell will become specialized, and it will lose three things. It will lose totipotency, it lose totipotency, it will lose pluripotency, and it will lose multipotency. Now, those are the three things that a stem cell will lose during cell differentiation to become the final liver cell, which has a specific function. Anyways, thank you for watching. That's the end of the video. This is a part of stem cells. It's a very small part of stem cells, but it's a very important part, and it's the basic understanding of what a stem cell is. Thank you.